Okay. So Eloise, are we all set to go? Have people come in?
We have 39 people online at the moment watching us.
Wonderful. Okay, welcome to everybody. We're just going to hold off a little while. There's several events happening all at the same time or just finishing off at the moment. So we're just going to wait a few minutes more for people to join us. In the meantime, we have some lovely graphics in our backgrounds, and you can have a look at these wonderful little images. Okay, so I'm watching the numbers of participants increasing. So we're at 363 right now, so that's great. We'll just hold off for a few more minutes as more people are joining us. Okay. Okay. I think I'll just have a little chat there about our graphic in the background. So this is a graphic that was designed by Ran in Iceland. And we met Ran back at the end of 2019, October 2019, when we all went to Iceland for a workshop. And we were so incredibly impressed with the images that she was able to translate from our discussions into a graphic form, that we've invited her to continue to be with us in terms of our world, in terms of our graphics that we're producing in the Marine Microbiome working group. So they're pretty wonderful images, very colorful and everything. Okay, I can see now that we're at around 71 participants, and we'll just hold off for a little while longer. We're expecting in and around 100.
So while we are waiting, I can say that Ran, the artist, she will be making a new drawing for the World Microbiome Day, which is by end of this month. It's still a secret. I understand Eloise has a kind of a call there, but yeah. So just looking forward to see what she will come up with.
Thanks, Ziggy. Okay. Well, I think it's we're good. We're in around 70 odd. So I think it's a good time to start. So I'd just like to welcome everyone to the group, to the session today. We have some exciting sessions planned for you today. And one of the things the group here would have liked me to say was, to start by defining the microbiome for people who are unfamiliar with what the microbiome is. So it's a term used to describe communities of microbes and their combined genetic material that have settled in a particular environment. So for the marine, that means anything or anywhere from the coast to the deep sea and sediments to communities of microbes in and on organisms, for instance, sea stars, whales, even seaweeds, and everything in between. So our panel sessions today will start with panel 1, sharing best practices, infrastructures and benefits. And that will be moderated by Guy Cochran. And then that will be followed by panel 2 on socioeconomic value, ethics and tech transfer. That will be moderated by Sean Robinson. And then finally, panel 3, Sparking Fascination on Marine Microbes. That will be moderated by myself. And at the very end then, Kelly Goodwin is going to give a roundup and highlights from the panels, and she'll close out with our pledges. But first, before we go further in, I'd like to thank and acknowledge my co-chairs in the AORA Marine Microbiome Working Group, Kelly Goodwin at NOAA and Sean Robinson at Fisheries and Oceans Canada. And both of them you see here today, and you'll be meeting later in the panels. And so I'd like to thank Craig McLean, Terry Schaefer at NOAA, Erin McPherson and Andy White at Fisheries and Oceans Canada, and previously Trevor Swertfager and Karen Davison. And then from the European Commission, DG Research and Innovation, John Bell, Ziggy Gruber, and Elizabeth Balsey. All of these people are the custodians of the Galway Statement on Atlantic Ocean cooperation between Canada, the EU, and the USA. We want to thank also the EU Horizon 2020 Coordination and Support Actions, the AORA CSA and Anchor, and the All Atlantic 2021 Forum organizers for this amazing opportunity here today to hold an event on the marine microbiome. We also want to thank members of the AORA Marine Microbiome Working Group. So Pierre-Luigi Buttigieg, Anis Lacourzière Roussel, Teresa Lethieri, and René Groben, Danielle Iudiconi, Jorn Smith, of course, Stefan Pezon, who's here with us today, Ziggy Bjornsson, and Lidor Erelson from Rannes. Now, Atlantico is an all-Atlantic project and is also one of the main organizers of this event. And a special thanks and call out actually to Eloise Trabut, who's at Stazione Anton Doran, and also to Erin Bertrand at the Ocean Frontier Institute in Atlantic Canada. It's really great. Thanks to you for helping us out. And then one more person here that I need to thank would be Fabiano Thompson at the All Atlantic Joint Action on Marine Microbiotechnology. Really sincere thanks, Fabiano, for your help in helping us to organize and promote this event. And one more, we need to pay special thanks to the All Atlantic Youth Ambassador and Mentor, Bochumelo Sandra Foma, and Teresa Lethieri at the European Commission Joint Research Centre, Inderjeet Singh Marjara at the Blue Bio Co-Fund, and Issei Haversdottir at Mattis for her kind efforts on communications for this particular event and helping us promote it. Now, to all of the volunteers, the speakers, the panelists, the moderators, a sincere thanks for your considerable time and efforts for this event, and to all of our participants today for joining us. I'm now going to hand over to Sigurdur Bjornsson, who's at the Icelandic Centre for Research called Rannes. Sigurdur and his colleague Lidur Erelsson have provided just quite simply incredible support, including financial support to our working group, the Marine Microbiome working group, and all through COVID-19. And they helped to keep us on track, moving forward, and actually kept us motivated. And sincere thanks to you, Ziggy, to Lidur, and to Rannis. And now over to you, Ziggy.
Thanks, Margaret, for your nice words. But let's not forget that you've been with us from day one and been instrumental in running this working group. Before that, a kind of a work package in Aura. So thanks for all your help.
Thank you, Ziggy.
Well, Marine Microbiome is the origin of life on this earth. So this is quite important issue that we are discussing here today. We have quite many participants that have registered, but they are not all shown up yet. But we can see that they are coming from 31 countries and from all continents except the Antarctica. So there is interest, widespread interest, and the interest or the gender balance of the people that have registered is 55 female, 45 male. So this is all pretty good. Some good numbers there. The agenda, as Margaret said, I mean, we will have these 3 panels and they will run one after the other. But the agenda and the name of the panelists are on our website, marinmicrobiome.org. So feel free to download it and you will see there the names, their short bio, etc. And of course, do not forget to download the roadmap. The roadmap is there also. And that roadmap was presented actually almost 2 years ago when COVID started in Brussels, beginning of February 2019. Well, we will be running these panels one after the other, and during the panel sessions, you can put forward your questions, and there will be polling. And by the end of the event, we will follow up with pledges that you as an audience can also influence, because this will be a part of our Slido. So we are using Slido, and of course there is a chat window in the webinar, but all this is run by Eloise at Atlantico. I think she is sitting somewhere south in Italy. And so she will be running the show, so to speak. So over to you, Eloise.
Thank you very much. So we have, in parallel with this, with the webinar, we have a session on Slido. So you can join in for the little practice question, and I can share this screen so you can see all the information. So if you go to slido.com and you can enter the code 363467, or if you have your mobile phone handy, then you can scan the QR code and it should take you directly to the event. Oh, it shows us who's there. So I'll give you just a few seconds to join in, and then I'll launch the little quiz.
It's going really well, Eloise.
Okay, I'll launch the question here. So the test question is, when is World Microbiome Day? Is it on February the 5th, June the 27th, August the 18th, or there's no such thing as Microbiome Day?
That's a neat question.
Right. Yes, and we have 65% of you guessed it right. So World Microbiome Day will take place on June the 27th. And actually the Marine Microbiome Working Group will be doing some events on that day. So keep an eye out for that. And with that, I think we will go straight into panel one. So I'm handing over to Stefan, I believe, or to Guy.
I think you're handing over to me, to Guy. So thanks.
Excellent.
Thanks very much for that. So welcome to the first panel, where we look at sharing best practices, infrastructures, and benefits. We have a number of initiatives that are represented in this discussion from within and beyond the marine sciences. And we have three leaders of initiatives. So first we have Dominique Auberton, who leads on marine oceanographic services, bringing many different services together. We have Catalina Lopez Correa, who is bringing together institutions and organizations in Canada to share data related to COVID-19. And we have Frank Muller-Erestrup, who is bringing together cities around the world to share their samples and their data relating to pathogen and microbial resistance. So what I'm going to do is first ask for introductions from the members of the panel. And I would like to ask first Catalina to give a brief introduction to herself and to the initiative. Then I'll switch to Frank. Then I will ask Stefan, as the organizer of the panel, to introduce himself. I will then introduce myself, and then we will hand over to Dominique, who will give a 5-minute presentation on operational oceanography in the digital twin ocean. So, Catalina, please.
Well, thank you, and thanks for the invitation today. So in terms of background, well, I have been working in the world of genomics in the last 20 years. Did my training in France and Belgium, worked for a little while in the UK, had the pleasure also to work in Iceland, in Reykjavik, with ecogenetics for about 4 years. Always mostly in human genetics, but since my arrival in Canada in 2008, I have been leading initiatives around the application of genomics in a multi-sector fashion with initially Genome Quebec in Montreal, then Genome BC in Vancouver, and now working with COVID. So on Genome Quebec, the work done in Genome Quebec and Genome BC, we had projects in microbiome, in fisheries and aquaculture. We had lots of interactions with Europe in those spaces, also forestry. So really application of genomics in a cross-sectoral fashion. Now, what I am doing with the Canadian COVID Genomics Network is, as Guy explained, initially we are actually Cancogen is called. This initiative was launched in April 2020 at the beginning of the pandemic. There was an investment of $40 million to sequence viral genomes and human genomes. So we have $20 million to sequence 150,000 viral genomes, and we have $20 million to sequence 10,000 human genomes of individuals with COVID. We have the goal and the role of Cancogen is to coordinate those efforts across different laboratories. For the viral component, we're working on a decentralized way, and I can explain a little bit more what challenges we're facing and what opportunities we're facing also in terms of data. So the viral component is decentralized, meaning that all the public health laboratories across Canada, we have about 12 different labs, public health laboratories, that are doing the sequencing, and then we are sharing those sequences and aggregating that information. On the human side, we have only 3 centralized facilities that are have been historically working together and exchanging the data. And the overall objective of Cancogen, of this COVID Genomics Initiative, is to use the data. It's not really a sequencing project. It's more, I would say, a public health and policy project, because the idea is to use this data to inform public health and policy decisions around COVID in Canada. So going really beyond the data, and that's why aggregating, sharing, and integrating data is so critical for countries. So I will leave it there maybe for the introduction, and I can provide more details on the question.
Great, thank you very much, Catalina. So Frank, over to you for your brief introduction.
Yeah, so very briefly, so I think that we are, from our side, involved in quite a large number of projects where we're trying to characterize and understand the microbiome in different reservoirs, but that's mainly the humans and the animal reservoirs, and very much looking at the infectious disease agents, whether that is antimicrobial resistance, viruses, or bacterial pathogens. And of course, in that aspect, to try to understand the transmission of diseases around the world, but basically we need publicly available microbiome, from everywhere and from all reservoirs. And of course, there's for the last, in the VEO project that I'm representing here, there's been a lot of focus during the last year on COVID-19 and how can we actually assist in sharing these data and also making what you call pre-analytic data available, because it's not everybody that might be able to call, SNPs or identify specific genes from these data. So also try to make some processing of the data available for everybody. And that's, of course, a nice combination of also meeting some of the different, legal challenges. I had just prior to this meeting been sitting for 3 hours listening to all the arguments regarding why Denmark are not sharing COVID-19 sequences. And it's actually our, the chief lawyer for the government that has stopped this because it's considered human genomes that cannot be shared right now. And it's incredible that it's not so much the technical things, it's very much, the legal, surroundings around the sharing of data that is getting into the way. But I think we'll overcome that. I think that one of the challenges that we see is that it's not only a question now of just getting more raw data yet, it's also that we actually need to consider also how can we pre-analyze some of these data so they might be accessible to more people. It's not everybody that can, pull down all the microbiome data for the world, and do assemblies and look for interesting genes. There needs to be something that is pre-analyzing this so we can really reach out to the epidemiologists and people that are not bioinformaticians. So I think that's covered it, Guy.
Great, thanks so much for that, Frank. So over to Stefan briefly.
Yes, very briefly. So I am part of the Aura working group. I'm also co-coordinating at Anteco. So it's really a pleasure to be here today and organizing the first panel. So I will be sitting in the background, looking at the polling, looking at the chat, and at some point I will interrupt Guy and come up with the results of the polling. So as soon as Dominique starts presenting, I think that Eloise will start our questions for panel 1. You'll have all 3 questions. So you can, while Dominique is speaking, while the discussion is going on, you can answer the questions and we'll come back to you with that. Thank you.
Thank you, Stefan. So just to introduce myself briefly, I'm Guy Cochrane from the EMBL European Bioinformatics Institute, where my group provides data services specifically around molecular biology data and sequencing data. We support many different domains of science, from human through livestock, but a lot of our work is around microbiomes. We've been active in standards and data coordination on the marine and the aquatic side, and also we're leading on the European COVID-19 data platform as well, with Frank and others. So we're involved in many different microbiomes. So with that, let's hand over to Dominique for an introduction and your presentation, please.
Okay. So can I have my presentation, Eloise? Thanks a lot. So it's going to be something very short as was asked, and hopefully it will open discussion and perspective on the digital twin of the ocean. So I've chosen, so my background is in physical oceanography, so I don't know anything about your world, but it's also for that, but it is very interesting. So I've been much involved in the past years in the Copernicus Marine Survey, so I would like to talk a little bit about that and beyond. As the time is short, I'm going to focus on a few points. So I've chosen the point because I thought they can be interesting for the marine microbiome. They led to many discussions between us and then to agreements. And then they help us to move forward on the service and on the access to the data. Can I have next slide, please? Okay, so I choose 3 points. So the first one is the access to the data. For up to now, for the environmental data and products, there is 2 possibilities of access. The first one is through the Copernicus Marine Service, when any user can discover, view, and download the data. The other possibility is through the Copernicus JAS, for example, as Wikio, the Wikio one, where the user can, in addition to discover, view, and download the data from the Marine Service or for any of the other Copernicus service, and as well as the space components, the satellite data. You can also use infrastructures, platform, and software as an e-service. And in the future, we will have, hopefully, the digital twin of the ocean that would allow to discover, view, and download the data as in the Copernicus Marine Service, with some expertise on the marine science, and also use cloud-based processing tools that will be installed and developed on the digital twin of the ocean. An important point, well, some points that are interesting to know about users, that usually they're keen with new development and modern accesses, but at the same time, some of them at least, they still want old access to data, for example, as FTP. Scientists, my experience is that scientists, they want to have state-of-the-art data sets and products. Other users don't. They are more interested by original area, by some parameters, by the period of time. But all of them, all the users, as soon as they access to an operational service, they want to have the access to the dataset and product to work, which means that the product needs to be available and on time. Next slide, please. So the second point I wanted to focus on is the simplicity of the offer. This is something that is important, and the offer needs to be as simple as possible, because it will be then easily defined and explained and promoted and so on. Knowing that the complexity of the offer will increase with time, with new data type, with new parameters, with new formats. So it's much better if it's easy at the beginning, than because the complexity will increase with time. And then the last point and the most important one is that the offer will be easily found by the user. And this is what we want. We want to have new users, other communities, to find the service and to access to the service. These three points are asked by the users, especially new communities, and a little bit outside the core and the usual community that we have as users. And it's all these points are also asked, and then we need to discuss and agree with the producers and the other data providers how to deal with that. So the offer, from what we know, what we have done in Copernicus, it's need to be seen as a whole. So it's a full catalog, only one catalog, plus additional ones that can be subset, that are usually subset of the full one, for some specific application, for some specific environmental data policy, for example, or for some specific communities of users. The offer also needs to be homogeneous. This means that it needs to have we need to have a common definition for what a dataset is, what a product is. We also need to agree on a set of metadata to characterize the dataset and the product. And these need to be consistent with for example, the European Open Science Cloud, EOSC, or other initiative, or European or international. And we need to use international standards and formats that can be completed or limited when necessary for the specific application we have in the service. And of course, the offer needs to comply with the data policy chosen, and that has to be followed. Last slide, please. And the last point I wanted to focus on, it's concerns the evolution of the service. So which and what evolution of the service? So the first, okay, there is a proposition of a service, there is an offer, and the user use the service. And then the best thing probably to do is to listen to user feedback and to criticisms. And then also, and using this user feedback, and then also to listen to producer and data providers for any scientific and technical improvements. We all together at the management team, you discuss, you agree, and you choose the next implementation to be added in the service to improve it. But knowing that for an operational service, the user don't want many change. Ideally, they don't want more than one change per year on their product they use. And they also want to have an announce of the change. Ideally, 2 years is a request to have to know about the change, which is a bit difficult to do. So that's it. I would just wanted to focus on these 3 points. But if you have any question on these points or any other question concerning the service and operational service, I will be happy to answer to the question. Thank you very much.
Great, thank you very much, Dominique. So what I'd like, I'm going to start the questioning, and we had very little time. What I'd like to do is pick up on a point you made on your penultimate slide about agreed metadata and about data standards. But I would ask a question, I think, first to Frank. What is it in the, perhaps in COVID, but also in the sewage surveillance work? What is the approach to being able to collect data from multiple sources, but still have data that across which one can interoperate?
Well, I think that the key is always, standardization. So, and especially if you want to use data for epidemiology, so we want to use a number from a given reservoir and compare that to a number from another reservoir. Well, then we need to have, then it needs to be the same, same sample matrix, and we also need to generate the data in a reasonably similar way, because otherwise we're really, disturbing the numbers, and we cannot really use it for epidemiology. So I think that's, the key there is basically standardization. The same goes also for COVID. That's right now it is mainly using qPCR and just looking at the numbers there. But if you run the PCR, the purification in different ways, you're also going to get different numbers. So again, standardization is key. It is not as critical if you just want to get the sequence and say, do you have this specific variant present there? And do you have this specific variant present in another way? So if you want to look at assemblies and to look at, the variants, then it's not as critical to get, the standardization. But still, we need to get information of, where is it, what is it from, when was it sampled, et cetera, et cetera. So these classical, metadata that also need to go along with the sequencing data. And I would wish we get more metadata, especially in a standardized way, because that will certainly help the downstream analysis.
Thanks very much, Frank. So I'll hand over to Catalina and I'll pick up that final point actually, which is I think the tension between the power of an analysis that requires a lot of metadata and a lot of integrity, and the complexity for the data providers to provide or to capture that and provide it. So what's across the Canadian network, Catalina, what what are the practical solutions to balancing data provider effort against the research outcomes that you can get from analyzing richly described data?
Yes, well, so on our pipeline for all the work that we're doing on COVID genomics, we basically started at different levels. We started with developing harmonized protocols, as Frank say, that we all generate the same type of data with the same protocols. We also harmonize and standardize our quality assurance and quality control pipelines. So we develop quality assurance and control pipelines so that everybody uses the same quality assurance, quality control. Now, on the metadata and contextual clinical data, that's where we're facing some challenges. Also Frank mentioned challenges around privacy and security, and sometimes even political challenges. So in terms of metadata, for sharing internationally, in sharing at a pan-Canadian level, we're using just minimum metadata and standardizing and harmonizing the minimum metadata. That has been a real challenge. We actually have a working group at Campugen that's dedicated to harmonizing and just standardizing these minimum metadata. And then we have the contextual metadata that's treated because it's sensitive clinical information and public health information that's treated just with, is with very restricted access to only a group that is directly working on public health. So it's that balance that we're trying to find between giving access to the scientific community in general to at least the genomic information and the minimal metadata so that some general analysis can be done. But even with that, we're facing challenges basically in 3 areas. One will be initially the technical and even capacity challenge. So some labs, public health labs, are just starting to do genomics. They might not have the tools, the software, the equipment, the expertise, and the capacity to share the data. And then we have the, I would say, more the privacy, legal, and ethical challenges, where privacy officers in some provinces in Canada will say that viral data with the metadata, there's a risk of breaching privacy for individuals. And so provinces are different depending on the province. There are different legislations in terms of the privacy and security of the data. And that's, again, another barrier for sharing. And the last one I would say is also a little bit of cultural challenge, the culture of, I think lots of the scientists in the academic world are used to share data at an international level. They're used to deposit their data in international databases. Public health officials are mostly used to work with the sensitive data, with their databases, with their local databases, instead of sharing. So we have performed a map of these barriers, and we're developing now a strategy to try and tackle each of them: the technical, the legal, and also the cultural.
Great, thanks very much, Catherine. So let's on that theme, and I guess so. Marine data, I think there won't be so much concern about human data protection, but there will certainly be sensitivities, some of them relating to culture, some of them relating to perhaps commercial interest, career, future career opportunity interest. So let's throw that back to Dominique. What kind of challenges do you face in oceanography with data release or data sharing from data providers that is suitably open enough for people to be able to discover and access?
Well, the different type of data we have we have in situ observation data, we have satellite data, and we have modeling data. So we need to homogenize all of them to have an easier access as possible for users. More do you want the user to come, more you need something that is very simple as an access. And years after years, you see that you see new communities coming and using the data, the product, not and different types. Some of them in some region, usually, and some parameters only, day after days.
And so for the data providers, what kind of issues arise with sharing their data for those who are generating the data?
Well, when it is operational, it's a bit heavy. It's lots of work because they need, as we have near real-time data, it's meant that there is a monitoring, near real-time monitoring, real-time monitoring to be sure that the data are accessible for any users. And but at the same time, there is this near real-time data. And there is also past time data with a long period of time. So it's a bit heavy and it's two different access of the data. And in Copernicus, for the Marine Service, the near real-time access is okay. It works. It can be better, but it works well. But I've heard that the users said that for the long time series data, as it is big volume, it is a challenge and it is still difficult for the users to access all the data, or the area of geographical area they want of a set of data.
Guy, with 5 minutes left, it's my turn to interrupt, and I'll ask Eloise to pop up the results of this. Let's go to the second question, straight to the second, if possible. So we have 27 people who have answered. If you want to answer, you can still influence the results. So we were asking to rank the following challenges about including marine microbiome data in the digital twin of the ocean. So that concept of developing a digital representation of the ocean data where we can dive in, explore. So let's look, it's pretty much even. So the first one is the capacity to fill gaps in the spatial and temporal coverage of existing observations. So the need for new observations comes next, equal with the third one, a capacity to model observations into useful digital knowledge. So useful maps, something that can be more than just observations, but could be derived indicators, for example. It moved, number 3 moved to number 2 now. To visualize, the capacity to visualize, access, and use the digital knowledge that we generate. And last, the capacity to analyze new samples and generate new observations. In the case of marine microbiomes, it's true that sequencing, for example, is becoming more accessible, less expensive. So that's reflected in the answer. So I'll switch back to you, Guy, to pick up on these answers to continue the discussion.
So thanks very much, Stefan. So in the last couple of minutes, what I will do is project those questions onto the panelists. So I'd like to ask each three, just in a sentence, in order to include marine microbiome data into oceanographic models, so include alongside the data we heard about from Dominique, what are the top priorities? What are the key things we have to do? So just a sentence from each person. So Catalina first.
Well, I will say, based on our experience, I think is the capacity to harmonize and visualize, I think, are the two critical components.
Excellent, thank you. And to Frank.
So once again, I didn't get that.
So in order to integrate marine microbiome data with other oceanographic data to build models and to understand the oceans better, what is the top priority in a sentence?
Well, I do actually think it is the analysis and the integration. So I think that that's probably where I am at. But I can see that you're pretty close, all of you. So and also really that there's so many climate data, and that is actually just as large a data set, or even larger than the microbiome data, and the capacity to analyze that in a comprehensive way. That will be a major challenge.
Great, thank you, Frank. And then finally, the same question to you, Dominique.
Well, I would say we'll have to find a way to integrate them in the existing environmental data of the Copernicus service. with a name, with metadata, and so on for the user to be able to find either this data or the other one. So it's a big challenge, I think, but it's feasible.
Great. Thanks ever so much. So, well, we could go on for a lot longer. There's much more to discuss, but we don't have time, I'm afraid. So with that, I'd like to thank the panelists, and I hand over to Sean for the next panel session.
Thank you, Guy.
Great. Well, thank you very much. That was a great start. Welcome all to the panel on socioeconomic values, ethics, and the transfer of technology. I'm your moderator. My name is Sean Robinson, and I'm a research scientist who works with the Department of Fisheries and Oceans here in Canada on marine microbiomes and antimicrobial resistance. And I'm also chair of the Marine Microbiome group in AORA with Margaret and a bunch of other great people that are working with us. First of all, I want to thank you for taking the time this morning, this afternoon, this evening, wherever you're coming from. I know we've got a lot of meetings over the last year or so on Zoom, so I'm glad you're spending some of your time with us. We're going to talk about some pretty large topics here this afternoon. All of these are ones that could basically fill an entire conference session for, several years running. And so obviously we're not going to get into all the details of this, but we are going to give you a flavor of some of the things that you may want to be thinking about, or perhaps you already are thinking about for some of this. We're going to have 3 brief overviews from 3 panelists who are experts in these. And then we're going to have some poll questions for you. So you've already been through the polling with Slido. Feel free to try those out. It's really worthwhile, and will get us some data that we can use to not only for us, but for other groups as well. Hope you find this thing stimulating and enjoyable. There's also an opportunity to send in questions now, or you can put them into either Slido or send them into the chat and direct them either to panelists, or we'll answer them during the session, or we'll answer them afterwards. But the point is to try and get this whole conversation going. Try and put out some different themes that perhaps you're not thinking about. But with this new technology, this isn't a new iPhone coming out, a new iPhone model. This is a technology that's going to change the world for the next 100 years. And so it's worthwhile putting some thoughts into that. So let me get started. I'm going to introduce our 3 panelists in the order of speaking. You can see their detailed bios on the website if you haven't seen them already. The first is Chris Bowler from the Ecology and Evolutionary Biology section at IBNS in Paris, France. He's got some interesting things to tell you. Diogo Chochka from the Kopp Institute of Biology at the Universidade Federal del Rio de Janeiro in Brazil. He's a biomathematician and has some really interesting insights into the global south. And finally, batting cleanup, we have Aaron Bertrand from Dalhousie University and the Ocean Frontiers Institute, which is one of the leading organizations here on the East Coast, which is synthesizing a lot of different disciplines together. So with that, we're going to go ahead. We'll have a quick, brief presentation starting with Chris, then we'll go into some questions, discussions, then we'll transition to Diego and then to Aaron. So take it away, Chris.
Okay, good day, everybody. Eloise, can you put up my slides, please? Here we go. Great, thank you. So I'm going to give a brief talk about development and spread of technology in this session. The next slide. I think it's fair to say that our appreciation of the marine microbiome has really been driven by advances in DNA sequencing over the last decade or more. That's really because the DNA sequencing has allowed us to see what is invisible to the naked eye, right? You need technology to be able to see these microscopic organisms, and DNA sequencing has allowed us to do that. Now, this sequencing technology has been driven largely by biomedical research. We've seen that with COVID in the last year. And that's sort of the end of the story of DNA sequencing, if you like, because DNA sequencing over the last 20 years, since the technology has been used routinely, we've seen the costs go down massively, something like 5-fold reduction in costs. And at the same time, miniaturization of the machines have gone down as well. So we've gone from these big bulky machines down to little DNA sequencing machines that are, the size of a USB flash drive sort of thing. So that technology has really pushed things forward massively in marine microbiome research. The bottleneck now is really beyond DNA sequencing and is now at the level of bioinformatics, analysis of the data, and in terms of functional studies. So given that, the majority of genes that we find, certainly in the ocean, are of unknown function, we need to figure out what they do. And that still takes some time and will continue to take some time in future years. So that's a challenge that remains to be done. But as the technology is increasing, we can get down to miniaturization of the system. This has really revolutionized ocean research in terms of the ocean microbiome. We now have hundreds of millions of genes from hundreds of thousands of different organisms, be they bacteria, archaea, single-celled eukaryotes, and marine viruses. All of these are in the public domain and openly available to anybody who wants to explore them. The technology is now moving from sampling on ships and sequencing on land to sort of now doing everything on board ship because of the small, low-cost sequencing devices that are now available. The next slide shows us sort of as we move into the future, we can maybe envisage this sort of scenario where we have autonomous sensors that are doing the DNA sequencing in situ in the water column for us and beaming up that DNA sequence by satellite back to the labs on land for analysis. So the next slide sort of lists a series of challenges and opportunities that I wanted to flag. I think all of these are important. We need to learn how to bridge genomics with other marine-centered research to discover new phenomena and principles. That marine-centered research may be oceanography, it may be marine biology, it may be ecology, may be related to climate change, may be related to fisheries, aquaculture, or basic evolution of organisms, right? We need to learn how to integrate DNA sequencing-based technology with ocean observatories that already exist. And we need to learn how to integrate that data into ocean models, as Dominique has already alluded to in the previous session. To be able to do this properly, we need to establish operating principles and standards that everybody uses. And the opportunities really go from the design, the generation of new products and services through a range of different approaches. And I mean organisms, molecules, genes. Organisms may be a new organism for biofuel generation. Molecules may be new antibiotic. Genes may be new genes for plastic degradation, okay? Related to this, then we need to train a new generation of genomics-enabled oceanographers. And with all of this, we need improved international cooperation, benefit sharing for the whole international world. And with that, we need to think about ethics as well. And with that, I will end my presentation. Thank you, Sean.
Great, thanks, Chris. Perhaps, Eloise, you could launch the poll for this one now, please. So if you have any questions, please send them in. We're happy to field any of those. We have a couple minutes for Chris to answer any that you may have, or from the panel, or from our audience, of course. I guess there's and the poll is going on, so you can see the polls that we have there. Chris, how do you see this with all of these different needs and deliverables that have to come in? How do you see the development of this going? I mean, who's taking an active interest in this right now? Because is it private companies that are working in the background because they see funding opportunities or financial opportunities? Or
yes, there are private companies, but I guess it's largely academic scientists such as ourselves. there are individual groups that are thinking about this, like Aora, right? Thinking about how to standardize things. There are discussions in progress, but it does take time to figure out how to deal with new technology. When molecular biology first came along in the 1980s, there was initially a moratorium on using, recombinant DNA, as it took the scientific community a while to sort of establish, well, how do we want to do things? We may have to go through a similar scenario in marine microbiome research for certain applications. I think we're going to talk about this later in the session. But I think we don't really have structures in place right now to allow us to establish good operating principles at an international level. That would be my short answer, I'm afraid. So we really need that.
Okay, great. There was a question from Christine Liu, apparently. I can't see that on my machine here, but maybe we can copy that to the chat, or maybe I'm just looking at the wrong thing.
Yes, Sean, the question is for panel 1, so I'll put it in the chat for everyone, and I'll answer Christine.
So, okay. So I guess the next thing, let's move on to the next talk. We'll have some of this and we can come back to some of this if we have time. So the next talk is going to be by Diogo Turca, and he's going to be talking about the transition. Anyway, we're getting some interesting results on the surveys for this one. Okay, so Diogo, can you take it away, please? And perhaps, Eloise, we could start the poll as well for Diogo while he's giving his presentation.
Yeah, sure. Thank you for the inviting. Good morning, good afternoon, or good night for everyone who's watching us. And thank you very much. Please, Eloise, can you share the slides, please? Okay. And so I will talk a little bit about improving outcomes in the Global South. I am a professor at UFRJ, Brazil, Rio de Janeiro City. Next, please. I will talk about using the One Health vision, because when we're talking about improving outcomes in the South, Global South, we need to think and we need to remember that the concept of One Health, because everything is stick together. If, for example, we have problems in South America, it's maybe its problem can reflect on South America and even, of course, in North America or in other countries and other parts of the world. So knowing and sequencing and understanding, for example, the microbiomes of the Global South, Atlantic South, and not only, of course, Atlantic South, it can be a first step or to start and knowing how, for example, how we can integrate this data in the oceanographic models and so on. So this is why I first talk about improving outcomes, I need to remember that my presentation was there are beyond the idea and the concept of one vision. For example, we are all together in this world in the same Earth planet. Next, please. There is also an interesting case here in Brazil. For example, we have the new biome, call it, because we have some very interesting structures beyond the Amazon and in the end of the Amazon River. When we join in the sea, it's an oil region, and there is some studies there, but this is a new one that we need to preserve, and there are some studies in this region concerning microbiomes also, but we need more. What we know is only a piece of the global of all the picture. So this is why I see it's showing this slide. Next, please. And to do that, how we can access, how we can know, for example, the microbiome from the oceans or the South Atlantic. Here for us, sequencing, it's not so cheaper. It's very expensive indeed. And so we need to think, and we need to find ways to share. And I think that Chris' talk, it's very, very good, very interesting, because there are a bunch of initiatives that can facilitate the access to sequence, and also for sequence regions and some kind of material that it's mandatory for our studies. Next, please. And also, it's also very important here for us is the training courses and sharing bioinformatics tools, because here, especially in Brazil, there are some groups that work developing bioinformatics tools, but we are mostly consuming these services. And when we change and we exchange and change ideas and apply into a lot of training courses, for example, we can understand why if we rain too much in the Amazon forest, for example, how can this event impact on the microbiome from the South Atlantic, for example. So there are many initiatives also here in Brazil, but we need more also. Next, please. As for example, for the last 10 years or a little bit more, there are some initiatives like comparative microbial genomics and taxonomies. They are funding, but many of initiatives here in Brazil. And we are walking, but we need to maintain these investments to get something more cohesive and get something more interesting also to see the big picture that we are trying to resolve here. Next, Erin, please. Luis. And I guess, I don't know exactly this, but I guess this is an African proverb that say, if you want to go fast, go alone. And if you want to go far, go together. I think that is proverb. They say and they explain how together we are stronger than if we are working alone. So this is why the exchange of human resource and mutual research projects can help us to understand not only for the South America, for example, but for all the world, because, for example, a bacteria and a virus don't know borders, don't know country borders, as we can see with the COVID example. So how is my time? Okay, so this is why I propose, for example, is how can the South, the incoming global South can be helped by other countries and what more. Next, please. And of course, there are some very interesting initiatives like the All Atlantic Auction that we're talking about now, and others initiatives indeed here in Brazil. Our government are investing some money, but we are trying to manage, of course, and there is not an easy time for us here, but we are trying to do our best with what we have now. I guess this is the last one. Next, please. Yes, and I think that the main message is also everyone is part of the problem and the part of the solution. Okay, and this is our question that we.
Well, that's great, Diogo. Thank you very much. So I think Diogo's raised a lot of very interesting questions there that also seems to reflect a lot of the goals of the overall All Atlantic meeting, where they're looking for pledging, they're looking for linkages between the different networks as well as the different countries. And I think the poll question that came out was pretty definitive that there should be a more global structure for development of omics and ocean technologies. So there's a bit more of a survey here. So is there any other questions that are out there that people would like to ask, or from the panel, or points to be made, or from the audience? if we can, we're doing a good job on the polls. So international organizations are one with a poll number of 1.
100%.
Yes, exactly. Okay, so perhaps we should go on. You can continue putting the polls in as you go through. It's interesting there where the thoughts that people are having, and people are obviously thinking and have some ideas about how microbiomes should be developed and whatnot. Our next talk is going to be by Aaron Bertrand. And Aaron is going to be talking about some ethics that go on there and about some things that you may or may not have thought about. And she's going to be directly using polls. And, you know, so it would be really good if you could participate in this. And I think anyway, I'll leave it to Aaron to show you what she's got in mind, but I think you'll find it interesting. Take it away, Aaron.
Thank you, John. So Eloise, when we get a second, the slides would be great. So I'm going to be asking you all to weigh in on a series of hypothetical scenarios as we go through the slides here. There we go. And so my expertise professionally is really in trying to understand how ocean microbes function and what that function means for biogeochemical processes in the ocean. But as part of my teaching responsibilities, I think a lot about ethical issues, especially those that might be arising from our efforts to leverage marine microbiomes to try to do things that are useful for us. So I'll take you through some scenarios. All of them are hypothetical. Some are more hypothetical than others, and you'll see that. But I'd like you to weigh in on them and tell us what you think as we go through them here. So the next slide, these are some issues that I want us to be thinking about as we go through our scenarios here. So in terms of risk and uncertainty, for each scenario, do we know enough to predict the outcomes with an appropriate amount of certainty? What kinds of outcomes do you think we should be considering? Should these be just environmental outcomes? Should these be straight economic outcomes? Sorry, go back, Louise. Or should we also be considering socioeconomic justice type of outcomes? How much uncertainty in each of these is too much? We also need to consider in these scenarios issues about informed consent and transboundary considerations. So who gets to decide? Who makes these decisions about how we put marine microbes to work? And at what stage does everyone get to weigh in? Okay, so let's think about our first scenario on the next slide here. So in our first scenario, university, government, and industry collaboration has led to the identification of a particularly rare marine microbe that degrades oil really fast, much faster than most naturally occurring marine microbes. So should we say there is a major oil spill off the coast of Nova Scotia, my home province, should we seed the ocean with this rare microbe in order to alleviate some of that oil spill problems? So you can weigh in on this question in Slido. And we don't have to look at this just now. We're going to look at them all at the end. Sorry, Eloise.
Sorry, I have to kind of stop sharing to open the question at the same time. I just realized that, sorry.
Yeah, so we have very simplistic answers, yes, no, and I'm not sure for this one. And then our next scenario is just a slight modification on that one, where this isn't a naturally occurring microbe that we've decided we could potentially use for oil spill remediation, but instead it's a microbe that we've engineered to do that. So should we deploy this engineered microbe in order to alleviate some of our problems from this large oil spill? So those are two scenarios, and let's move through to another one. Oh, sorry, Eloise, I didn't realize this needed to switch back and forth. That's annoying for you.
We're putting her through her paces anyway.
But this gives folks plenty of time to think about this and weigh in, so that's good. All right, in our next scenario, we have an industry group that has developed a genetically modified phytoplankton strain that produces a particularly valuable bioproduct that until now we could only produce through really small scale farming operations. And those small scale farming operations mostly take place in the global south and are pretty important. So a question we can ask ourselves is, should we be scaling up industrial bioproduction of these engineered algae? And our last scenario, one that I find particularly interesting. So we've got a situation where an academic industry collaboration has developed a genetically modified phytoplankton strain that sinks faster in the ocean than naturally occurring plankton. And they can spread that ability to sink faster to other phytoplankton they encounter in the ocean. Because of this ability to sink faster, modeling studies suggest that if we release this strain into the ocean, this would increase the amount of CO2 that the ocean removes from the atmosphere by up to 20% by the year 2100. So should we release this phytoplankton strain into the open North Atlantic Ocean? Well, this is our last Slido question for this ethics session here. And maybe Eloise, you can put up the answers to our questions and we can discuss here.
Just giving a few more seconds to answer the last one, and then I'll go back to the first one.
Great, thanks very much, Eloise. Yeah, so I've selected these scenarios to highlight some of the important ethical issues I think that we should be confronting when we think about many applications of marine microbiome derived technologies here, but these are picked out to be provocative and get us discussing. So you can feel free to weigh in the Q&A here. But this is what I was sort of hoping to see, that the I'm not sure answer is a dominant one. So I'm interested to hear from other panelists on these as well. If anybody wants to weigh in.
So this is good. I mean, this is giving us some real trends that that are showing a number of things. So we have 2 minutes left in this session before we wrap it up and hand it over to panel 3. But I guess I'm pleased with the way it's coming, and I'm really pleased the way the audience has sort of jumped in and contributed to this. I mean, I see a lot of conservatism or hesitancy about just jumping into new technologies here. I also see a bit of universality here, and a lot of the responses have been that more towards a collaborative view to developing microbiome technologies and databases and information and the resources that go with that on more of a global basis, which fits into what Diogo was talking about with regard to the One Health concept, where, I mean, we're all linked in this together. And it also fits into this All Atlantic meeting where we have senior ministers talking about trying to cooperate. I mean, there's desires for the EU and Canada, the US, Brazil, Argentina, Africa, all to get together to sort of collaborate and produce some of this information. So I think these are obviously questions that will dominate, I think, some of the discussions for years to come as we come to some solutions for that, and it'll obviously impact some of the younger generations as well. So coming up to our time frame, I guess I would just like to say thank you for your participation in this. Thank you very much to our 3 panelists, Chris, Diogo, and Aaron, for putting this together and putting some provocative and some thought-producing some presentations together. And with that, I will turn it over to Margaret, I guess, in panel 3. Thank you.
Thank you, Sean. Certainly very interesting, Aaron. Finish there. I must say, very provocative questions. I smiled immediately when I saw them. Okay, we're on panel 3 now, which is all about sparking fascination about the marine microbiome. And I would like to introduce Henk Bulhuis, Sandra Butumelifoma, and Astrid Schrader in this panel. And I would like the panelists just to give a quick introduction to themselves. And I'll start with A for Astrid. So Astrid, if you could introduce yourself.
Thanks so much for the invitation to be here. I'm kind of an outsider to this whole event. I'm a social scientist. I am studying scientists who study marine microbes. So I'm a lecturer at the University of Exeter, and I'm work in sociology and philosophy, but my field is actually called science and technology studies. So you are all my objects of study.
That's very provocative in itself, Astrid. Very good. Okay, thank you. Henk, can I move to you? I'll do things alphabetically. Can I move to you now, Henk, to introduce yourself?
So yes, my name is Henk Bolhuis from the Royal Netherlands Institute of Sea Research. I'm a microbiologist for already over 30 years. And I think you will learn more about my fascination during my presentation in a minute.
Excellent, that's great. Thank you, Henk. And to Sandra, can you introduce yourself?
Hello, everyone. I'm Sandra Poma, coming in from South Africa. And I am currently doing a PhD in ocean microbiomes, actually, which is exciting for me here to be here because I get to put some things on a literature review. And I was part of the All Atlantic Ocean Youth Ambassador cohort, the 2019 and 2020. And now I serve as an All Atlantic Ocean Youth Ambassador mentor. So I'm happy to be here and happy to share my input.
That's great. Thank you, Sandra. I should mention as well that as with all of our previous panels, we will have Slido questions popping up. And if people could answer those questions as we go along, that would be terrific. So first off, I'm going to hand over to Henk, who has a beautiful presentation ready for us. So Henk, it's all yours.
So Eloise, if you can start it in 1 second, please. Yes, now. Because this is a movie and I have to carefully time the number of words that I have to speak for every slide. So this is about my fascination, and I hope I can spark some of that fascination for the marine microbiome to you, the audience. Little did we know about the unseen majority until about 350 years ago. Antonio Vallejo gave us a first glimpse into the microbial world. And we currently know now that about 90% of the total biomass in the ocean is microbial. To give you some numbers, there are about 10 million times more bacteria in the ocean than there are stars in the universe, and many more than there are sand grains. So we have new imagery, we can make beautiful pictures. This is, for example, Coleophysiculus, a cyanobacterium. But we could already see the bacteria from the sky. We could already see them by bare eyes, especially when they grow in large mass, like in these algal blooms or in the red tide by dinoflagellates. And here you find these very unique fairy rings on microbial mats. Again, microbial life. Also, we have found now recently very large microbes that can be seen by the naked eye, like Theo margarita. And we now know how important the microbes are for the element cycles in nature, for producing oxygen, but also for fixing nitrogen. So in the end, it's a huge complexity of microorganisms, of different systems, of different potential, what it can do. And for that reason, let's call planet Ocean, planet Earth, let's call it planet Microbe. So the microbiome is already introduced by Margaret. And indeed, the marine microbiome is a collection of many, many different ecosystems, many different sites, many different organisms that all have their own specific microbiome, from the surface to the bottom of the sea, and also the deep sea with its unique organisms. We find it in polar regions, we find it in the tropical seas, the mangroves, sargassum, but also on ulva. There's a seaweed that have their own microbiome. And of course, also on fish and in the intestines of fish, all have their own microbiome that is part of the marine microbiome. Even on the grain of sand, there can be up to 1,000 different bacteria. And unfortunately, also the plastic microbiome that we created more or less ourselves. Again, a mind-boggling, enormous diversity. So within Europe, we have 4 projects dedicated to microbiome research for sustainable food production, and I'm part of SIMBA. And SIMBA has a work package for the marine microbiome, and we look at the use of microbiomes for better quality of seaweed, algae, fish farming, but also of the saline agriculture. And you have to imagine that potatoes, as you can see here, they grow very bad when we increase the salt concentration from left to right. And what we're trying to do now is to grow these potatoes under different salt concentrations, but we amend them with the microbiome from a natural salt marsh, there where the microbiome is already optimized for the salt concentration. Unfortunately, I cannot yet tell you the results. We have to wait still 2 months before the results are in of this potato, but they're growing well on the salt concentration. That's cool. And so how can we spark fascination to a wider audience? I mean, too much of the microbiome is still associated with disease, with food spoilers, while in the end, they are producers of a lot of medicine and they can help fermenting food. So they are also preserving food. A very beautiful way to look at microbes is the new Microbial Zoo, the first and unique Microbial Zoo, Micropia. This is in Amsterdam, and it's absolutely unique. It's educative. You can go there with your kids, and you have many living microorganisms on display. And I could all invite you to go. Here's a wall of hands. And they also have a very nice website that you can look up for more information. I was happy to be part of a project, an exhibition called Biofilm by Wim van Egmond, a photographer. And there we studied the biofilms in microbial mats in the sediments of beaches. And in here you will find a very beautiful layered structure in which the different colored layers are formed by different types of bacteria, the purple sulfur bacteria and the cyanobacteria. And again, beautiful pictures we obtained from this ecosystem. Here we have euglena and algae, and also some movies. And you see a diatom with cyanobacteria. And so, yeah, in the end, as a scientist, we have to get these schedules and see which kind of interactions there are. And we discovered that part of these organisms have a rhythm. They have a day-night rhythm, a biological clock, but it's not fun to look at charts, I think. It's much better to see it in action. And this is what we see here. Here we have different movies of the day-night rhythm of bacteria that go up at night and down during the day. And that's caused because during the day, the algae produce a lot of oxygen. And during the night, the oxygen gets depleted and the bacteria have to go up to the surface to get to the required amount of oxygen. And they form these amazing, intricate nodes. It's almost like a neural network. It's absolutely interesting to see these patterns. This is our time-lapse movies, all done by Wim van Egmond. And this is what you see if you look on an ecosystem from the top. This was actually done in the field, so we have to be careful for high water coming in. It takes a little bit. And we also now use it for experimentation, because here, for example, we see how the algae slowly oxygenate the sediments, and you can see the bubbles forming at the back. Also, microbes themselves, they can be a source of inspiration for artists. This is the square haloquadratum, halophilic archaeon. You see computer images, you see a nice ink drawings of this organism. And I've recently discovered that somebody made a filled square. There's a student that I'm supervising is from the art school, and she's actually making a contraption, a construction with plastic chambers filled with algae And to fix your own carbon at home. It can be driven by LED panels, by LED light, or you can hang it in a window in order to receive sunlight. So absolutely nice ideas of these young students. I absolutely love that. So we also have to spark fascination to the stakeholders, the policymakers, the industries, the NGOs. And yeah, this is why we came to form Aurora. Aurora was already formed, but we combined forces. We went to the Decade of Ocean Science in Halifax. We mentioned the microbiome a lot, but we didn't make it to the mood board yet. And this was last year, but also a month later, we went to Brussels, where we had the All Atlantic Conference, and there we were very successful, especially thanks to a talk by Kelly Goodwin, that you will see later. So now we're officially Marine Microbiome Working Group, and we have this beautiful Marine Microbiome roadmap that I advise everybody to look at and to see what we've written for future research. And in the end, yes, again, there's the website called the Marine Microbiome.org. And finally, how can we spark more fascination? We have to take the franchise of movies as Jurassic Park and Star Trek, the astronomers and the paleontologists, and to see whether we can also create our movie and to promote the marine microbiome, but then with a probably better, more sexier movie star. This is it.
Thank you so much for that, Henk. Wonderful imagery, amazing those little videos of, you know, the oxygen levels and, you know, the activity during the daytime. Incredible, really fascinating. Okay, I'm conscious of the polls that we also need to do. So I wonder if we could put out one poll now, Eloise, for the first question on Slido. And in the meantime, I'll ask a question to the panelists. Why should we care, or why should you, the public, care about marine microbiomes? And maybe I could start with Astrid.
Yeah, I thought the best way to answer is maybe how I came to care about marine microbes, not so much of microbiomes, since I'm still trying to figure out how to care about the entire microbiome. But so there are two stories about how I came to care about marine microbes, a professional and a personal one. So I actually encountered microbes who are largely invisible for the first time, swimming in them in the ocean, and those were bioluminescent dinoflagellates that glow up they spark and actually glow at night when you swim in them upon touch. So that was a very awe-inspiring experience, and that was about 20 years ago. And from then on, microbes never left me. So I tried to figure out why do they glow? Who are these? Those were dinoflagellates. I got into researching harmful algal blooms, and not like scientists would research harmful algal blooms. So as a social scientist, I ask questions like, how do scientists determine what is harmful and to whom in harmful algal blooms? So I was more interested in what kind of experiments do scientists do in order to determine that something is harmful to somebody else, right? And harmful to humans is different than harmful to ecosystems. So those were the kind of questions I kept asking from on the road along. The professional interest in the marine microbiome for me and why I care tremendously about all the research that you are doing here is, I think that metagenomics research is kind of contributing to a new conception of life. And this is my philosophical interest in the marine microbiome. You look at no longer at individual organisms, but as connections and relations and interactions. So, and this is a completely different view of looking at life, and also changes that view, changes what we actually care about and how do we care. Yeah, I think I leave it at this for now.
Can't hear you, Margaret.
Thank you so much. Okay, I sense that wonder, light, personal experience, and the kind of whole visual side of things, really fascinating. Can I pose the question also now to Sandra?
I think for me, it's very personal. I would say that it changed my life. I care about microbiomes because they put me in stages that I never thought it was possible. I was able to progress personally and professionally through researching the unseen majority. And I think back a lot about being a young girl, not understanding a lot about microbiomes because of access to information about them. So I care about them because I'm able to change other young kids' futures and trajectory in terms of their research and in terms of interest in science. And to do that, I've been able to work with NGOs to incorporate microbiome research on current curriculum, particularly for marine biology. So in South Africa, we do not take marine biology, is not part of natural sciences. So you learn about oceans when you are in university. So it's very unfortunate that that access to information does not happen at the earlier stages. I mean, we do know about Disney, we do know about that there is microbial life and as well as ocean life. However, as an inland girl, it's very hard to conceptualize what an ocean means. And so it's been, for me, a personal journey that I'm very grateful about. And I share much about microbiomes and how they touch my life.
Wonderful. Thank you, Sandra. It's very interesting to hear about, the great divide between, I suppose, the coastal side and the interior, I guess, and then also, the divide between primary, secondary, and university. Very interesting. Henk, can I also pose a question to you now?
Sure. I do a lot of metagenomics and bioinformatics, but I actually use it only as a tool because I'm interested in the ecology and evolution of ecosystems, especially of complex microbial ecosystems. And if you just realize that life originated in the ocean and that for about 3 billion years, the whole life on Earth was purely dominated by prokaryotes. And that the fact that the eukaryote cell is actually nothing more than a symbiosis of different bacterial and archaeal species that join together. I mean, in the end, we have to realize that we human beings and every other being are just walking mega marine microbiomes with the microbiome running through our veins. So I think it's very important to realize the connection that we have between us, between the whole Earth, and also the effects that the microbes have of the whole ecosystem. They just do not only affect life in the ocean, they affect the coastal regions, they affect the climate globally. So it's, for me, very important to study these microbiomes and to show that they are important.
Excellent. Thank you, Henk. It's fascinating that she's even, flowing through the veins. Wow, fantastic. I have another question here, and it's really from an all-Atlantic perspective. There have been so many different agreements signed now between the EU and Canada and US, and then also between the EU and Argentina, Brazil, Cabo Verde, and now obviously also South Africa, and now also with Morocco. So, Sandra, how can we apply, all the science diplomacy and, these Atlantic cooperation agreements made over, I guess the last 5, 10, 15 years to spark fascination about the marine microbiome?
Okay, so the thing about really these policy agreements and statements, for instance, this side event is a beneficiary of those existing policies. So they act as almost like a curriculum for us to be able to make informed choices on how we progress ocean research and innovation. So the Galway Statement on Atlantic Cooperation, which is pretty much I'd say that the initiator was between the EU, Canada, and the USA. So this really is a North-North framework that leads Atlantic cooperation. So that will mean that it drives innovation and research between those countries and the EU. So what came after? A lot happened between the Belém Statement and the Galway Statement, such as the South-South fragment framework, which was between South Africa and Brazil. However, the Belém Statement was on Atlantic research and innovation, a cooperation between the EU, South Africa, and Brazil. So that was more of a North-South framework that joins the Galway Statement, and then the other agreements that exist with Argentina, Cape Verde, and Morocco. So these really advance ocean research cooperation, capacity building, ocean stewardship, which just means that it's an ethical responsibility that everyone has to take care of the ocean, because it does affect the current generation and the future generation. But the most relevant mandate involves facilitating an engaged and ocean-aware Atlantic global community. So this is where we fit in, where we are using these frameworks to drive change, and we make actions towards how we can reach our communities. And like Diego said, it's really having a same vision, that we are all coming together to make sure that we have a healthy, clean, robust, and thriving ocean for all and for future generations. And this has to also include inclusion and diversity, making sure that nobody is left behind, particularly as well as the youth and indigenous communities, and how we make how we make any type of changes. So these also are just the umbrella that we're using here, particularly with our panel, which is Sparking Fascination. We'll use ocean literacy and science communication to reach communities, to inform them of the actions that they can make, to enlighten them of what the ocean's capability is, but also for them to be able to drive change in their communities by raising these issues with local governments, NGOs, as well as informing generations to come.
Okay, so I hear a common vision, shared resources, and a voice to government. Excellent. That's fantastic. I'm also now just looking at the poll. So the top 2 words coming through here in terms of what the marine microbiomes mean to folk here today is life and curiosity. It's nice to see some others here in sustainability and oxygen, undiscovered. Yeah, I think, for me, personally, the marine microbiome is that the undiscovered. It's the new era of discovery, yeah, I look forward to it. So I'm going to move on to question 3 for the panelists here. And if we could have maybe another one go out on Slido for participants, and please, participants, if you have any questions or anything like that, please put it into the Q&A. Okay, so I love this unseen majority. How do we enable the unseen majority to be appreciated? How to visualize microbiomes? How do we make them, known and appreciated? And one of the things there, I love the potatoes that Henk had, and looking at the microbiome, and how with flooding, coastal flooding happening, how can we still get food, from these areas? So that was really fascinating. And perhaps I could address that question to Astrid. How do we enable the unseen majority to be appreciated?
Yeah, thank you. It's a very difficult question. I think my point will be, if we want the microbiome, let me restart this. The difference between microbes and other organisms in the ocean is, of course, that microbes are invisible, that we can only get to know them either through their traces, that is, when they are harmful, to maybe the toxins they are producing, or the glows that they are producing, or the fish that they are killing if they do so. But we also visualize them in some way. And as many of us may have seen in the coronavirus was also a microbe, we have become very familiar with one kind of particular visualization. And those visualizations are not innocent. So it really matters how we, and I'm not an expert on visual media or anything. I'm just interested in how meanings are produced within the science. So, and visualizations is important there. I'm not trying to make a recommendation there, but maybe as a way to think about the reputation of microbes in the public. When you hear the word microbes, most people still think germs, and germs are agents of disease, and they mainly harmful, and we've got to get rid of them. But this image also came with a specific kind of technology, with a specific way of producing microbes in the laboratory. These were usually isolated strains, isolated cultures that were then isolated in order to infect other organisms. So this image of this germ, this microbe that we want to get rid of, is somehow related to how it was produced in the laboratory. Now, if we want to create appreciation for the ecological importance, actually that the microbiome is supporting our life and all life in the ocean. So I think the technology that you are all using, the public needs to understand and to appreciate how that is different from the old technologies that isolated these microbes, and appreciate the networks that you are detecting and also creating in a way. So and this is kind of what my field, science and technology study, is doing. It's looking at how technologies are making meaning of what you're finding. So so the point is that my personal appreciation is also very much influenced from the tremendous passion and appreciation I see from the scientists who are studying the microbes. So what we just saw in Hank's movie, there is so much already appreciation in there. So the question is how to transfer that into the public to get a greater appreciation from the public.
Thank you, Astrid. But that's really insightful, I think, and it's great to have somebody who's not a marine microbiome scientist looking at this and studying the scientists indeed. The visualizations I equally think so important to do. I wonder if I see we have here in one of the polls that how will you help us with a year-round campaign to spark fascination about the marine microbiome? Wonderful, folks. This is what we had hoped, that you would subscribe to our forum here and helping us promote through your networks and social media. Oh, wonderful graphics and images. That really goes to exactly what Astrid was saying here, the visualizations to help the appreciation, and also a lovely marine microbe to promote one per month. And folks also have some content videos and that type of thing. Wonderful. I think that really plays into what Astrid was just saying here. That's wonderful. Okay, I'm going to move on. Thank you, Astrid. And I'm going to ask my next question to Sandra. As an All-Atlantic Youth Ambassador mentor this year, and having experienced last year as a youth ambassador herself, how can we involve the All-Atlantic Youth Ambassadors? How can we get them involved in the marine microbiome and helping this appreciation happen?
Okay, so what I love about the youth ambassadors is that currently they are qualified scientists, qualified social scientists, qualified filmmakers. So we do have the skills, we are capable of being at the table, like as the youth people say, like create the table, make space for us at the table. It's because, you know, what motivates really including the youth is that half of the world's population is estimated to be younger than 30 years. So what is important is that however the population is really a substantial number, youth people are underrepresented in a global scale in terms of decision making as well as driving change. So, and I think a lot of the youth, even when I attended the Youth Ambassador side event, is that the youth are reiterating that they will have to live with consequences and decisions that are currently made by the generations that are before them. So it's important that the youth is included in any type of change and action that can actually motivate as well as drive any type of consequences that they will have to inherit. So when a lot of the times when I was a youth ambassador, it was really, everybody had this notion that youth ambassadors are added as an extra. We are the plus one to a lot of the ideas that come from policies and governments. We actually want to be there when you are thinking about an idea, when you want to drive change. Include us at the beginning because we are adding our experiences. We have a lived experience and we do bridge a gap between different age gaps. So for instance, with myself, I have access to my grandparents, access to my parents, as well as my younger generation. You know, I'm still cool enough to be on Twitter and be able to touch so many lives. So it's important that the youth isn't involved because, as I said, we are qualified. We are positioned to drive change because we are existing now and we bridge a gap between different generations. And we are open and ready to contribute. Margaret, you know, I always answer your emails. I think we're very much invigorated and so curious to add our voices because we know that our voices matter now. And it's exciting to be a youth right now because so many voices are being considered. There's so much representation and diversity. Everyone's voice really is valued because of so much work that is currently happening now. So we are more than happy to share responsibilities as well as accountability. You know, we have to really learn how to be ethical researchers right now. And through being in panels like this, I'm so inspired. And I remember when I was in Brussels, when I met the working group for the first time, I was a bit starstruck because I was like, oh my gosh, I cited this person, I cited that person. So it's very encouraging for me, you know, it's inspirational because I get to be part of the mix and be able to have mentors because you all have maybe, you just think that you're just doing your job, but you're really being exemplary in the way that I carry myself and the way that I push, you know, this microbiome work. So I'm very inspired and we are all inspired. And the current cohort, I saw that Marissa is here from the current cohort. So they are very much inspired and they do want to get involved. So please, when you need us, let us know. We'll be there. Thank you.
Thank you, Sandra. Absolutely. We do want to hear more from the All Atlantic Youth Ambassadors. We do want to hear more from you and from the mentors. Yes, we would love to get you more involved, and we hope to run a year-round campaign, as you know. So we're looking forward to that. I know that we probably won't be able to answer the polls just right now. I will move on. I had another question, but I'm very conscious of time moving on. So at this point in time, I'm going to pass on to Kelly Goodwin to do an overview of today and highlights coming through. And also onto our pledges. I wish to thank panel 3 for a really fascinating discussion. And I do want to follow up with you all afterwards. Okay. Thank you, and thank you to everybody who participated in our poll.
Okay, Eloise, I have been frantically creating a slide deck, believe it or not. I've been very busy. Can I share that or do I need to send it to you?
No, you should be able to share it yourself.
All righty, let's see if I can figure that out. Share screen. Okay, here we go. All right, can everybody see that?
Yeah.
Great. All right, so I had the task of frantically trying to ingest everything while everyone was talking, and I don't claim to have caught it all, but you guys will tell me what I missed. One of the things that we had to do as part of this is make pledges. And I'm going to basically summarize what we already said we would pledge and then open it up for you guys to add, each of you, all the participants, to add to what we're going to do for the pledging. This group will participate in World Ocean Day, June 8th, and World Microbiome Day on the 27th. And we encourage everybody to subscribe to the website, to download and inform and share people about the Marine Microbiome Roadmap, and actually read it. I was looking through it last night and I realized, oh my gosh, we have a lot of actions in there that we need to shepherd. So we should probably just pledge to think about that as a pledge to actually get those actions done. Then the year-round awareness campaign. You can see the hashtags. We have a LinkedIn page, we have a Facebook page, we have a Twitter page, which I guess you just can search for it and find it on Twitter. And then the idea is Microbe of the Month, scientist highlights, interviews, and engaging our youth ambassadors. Also, there is Marine Microbiomes Towards Standard Methods and Best Practices special issue in Frontiers. I assume we'll make this slide available on the webpage, and you can click on that, and please, the group, people from this group are editors, please submit an article. And most importantly, we are working to extend this group more broadly from the working group that was sort of assigned via implementation of Galway through AORA to what we're going to call a forum, to a larger group of individuals. And we need you to share your energy, join the team, spread the word, because at this point, no one's getting paid to do this. So this is a grassroots effort to make this happen. I also was assigned to sort of summarize what we had learned, what happened before. This is a very high level summary. Only 65% of people knew that World Microbiome Day is June 27th. Hopefully now 100% of people know. So that'll already be a success. In the Standards and Best Practices panel, I saw a commitment to incorporate the microbiome into that digital twin of the ocean. So I was super excited about that. Digital twin is something like a UN decade level action. To have the microbiome as part of the digital twin is no small feat. That would be huge. Poll results suggested that getting and analyzing the samples, including the spatial and temporal, is a big challenge. But there are also very large challenges in turning those observations into digital knowledge, as you might imagine, because microbiomes are big data. Panel 2, the socioeconomic value, ethics, and transfer of technology. I thought the challenges were well summarized. We'll put those slide decks on the website, so I'm not going to redo them there because they were so nicely summarized there. And then we got to experience many tricky questions that require answers, particularly regarding environmental release of engineered organisms. That is going to continue to be a big topic. I can tell you in the United States, there is an amendment based in Congress right now called the Bioeconomy Act that specifically has the words engineering biology in it. And a lot of those examples that we got today were engineering biology questions, which is, as agencies and individuals, we are really going to need to deal with that. So I thought those were great questions. Panel 3, I saw, put the two together. Microbes are curious life. So that's very, very, very cool. I think people got really engaged with the sparking fascination. I know I'm excited about that. It's a great way to bring people to our cause. And we do definitely need to work on that positive message that, microbiomes are not just germs. Thank you, panelists. CBio is here. Someone, I was texting someone, please send me the actual link to the BIOS on the page. I couldn't find it, so I bet other people couldn't. I also started a thing looking at our partnerships. I would like first maybe we can do this as a Google Drive for everyone who participated, who might want to add themselves as a partnership, who are all our partners. The Microbiome Working Group is EU, Canada, and the US. And Canada and the US are both from federal agencies, but we're really wanting to expand. So if you're out there and we need more participation, I think from Canada and the US in particular, many academic partners on our panelists. If you look on the website, there was a bunch of partners there. Genome Canada, and this was just starting to look at some of these partnerships, which I think are really important for us to capture, particularly for those of us who speak to leadership. This is the sort of things they like to see. So I think we should pledge to figure out how to capture this better. Looking at the poll questions, just to review, since maybe not everyone was looking at them, people were saying, yeah, I've looked for these data sets, I can't find them. And, oh, I forgot to put this as an action item. This group could actually put on the portal, where do you find data sets in microbiome? So I'll add that one in. The very valid point that sequencing is still expensive for developing countries. We talk about how much it's come down and how sequencing breaks Moore's law and blah, blah, blah. But that is a very, a viewpoint that comes from a northern perspective, if you will. And so this is something I think we should take as maybe one of our very first things to do on World Microbiome Day is in our social media campaign to note that disparity and moreover, see what we can do about it. We get questions about, hey, we've been talking about standardization for a long time, decades. What's gonna break? What are we gonna do to fix it? Questions about the digital twin. It's a view. It's not going to be perfect. Who controls it? How do we specifically train the next generation of microbiome scientists? So action items I got for this group, on our portal, what are the training opportunities? What are the seminar opportunities? How do we really not only bring, media coverage to the disparity? Now that we are all Atlantic, I think this is super important. You're telling the US diversity, equity, and inclusion is huge. And I am really looking, you can email me, kelly.goen@noaa.gov. You can email us if there's a contact us on the bottom of the webpage to give us ideas on specific things we could do. Most of these things require money, sure, but I have to, what are we asking money for? And then also the portal on finding databases. We have 3 accepted pledges. I'm not going to go through them because you can see them here. But if you go to the pledge list, there's some pretty graphics. I did them at the 11th hour last night. I didn't realize that we had to enter our pledges in this specific format. But we have 3 all Marine Microbiome Working Group expansion. And that's sort of all those things that we're going to do that I already talked about just a few minutes ago. The only one added on here that you didn't see is facilitating communication regarding national strategic plans. So if anyone's online and wants to work on a national plan for eDNA or omics or microbiome, the US has one, Canada's developing one. We'd be happy to work with you. Then the 2 other things, food security, ocean resources, and biodiversity. They have pre-labeled things, map and quantify the DNA of the Atlantic Ocean. Yay, we want to do that. And understanding the role of biodiversity in maintaining marine ecosystem function. And then I had to put in some stuff in there. And one of the things that's coming out of this is for Atlantico, the US is putting scientists on some Atlantico missions. So that's something we could pledge. One that we have to work on, exploiting the potential of marine biotechnology. This is just lifted directly from the roadmap. You know, and I basically said we're going to raise awareness, because right now without money per se, you know, what can we do? Well, we can raise awareness and try to generate funds. But pillars of the roadmap are biodiscovery and food value chain, and that fits right into that marine biotechnology. And then also there was some stuff now that came out from our discussion that I would have put in, but this is what I said we pledged, and we should see this tomorrow. If you're going to show up tomorrow, there's a session that will review the pledges. Good news is these pledges were accepted, so we will be featured. That's it for me. I'm going to stop sharing, but now I'm going to, I'd like to hear what additional pledges you all have. And that's to the panelists and to people added to the Q&A, and in addition to the stuff we might take from the Slido. So you saw what we said we were going to say. What else are we going to do? What other pledges can we make? I'm not sure. Let's see, Q&A. There's one that says, I don't know if everyone can see that, talking about the future challenges, opportunities, partnerships. Will the Marine Microbiome Working Group try to foster brainstorming, networking around Horizon Europe? Yes, we actually have started discussing this. It'd be great. Eloise, can people talk? Can somebody actually break in to where they can participants now speak? Or can they only
they can't, but if they raise their hands, I will be able to unmute them.
That would be great, because I would, you know, it'd be really nice to have a conversation with people now, because I would love to hear what your idea on this specifically is. I can tell you one of the things we've discussed, you know, one of the problem with Horizon Europe is, for example, the US, you know, can't play. We can only do things in parallel. If you can't see what I'm talking about, if you go to the Q&A, you can see that this question is up. If you don't know about the Horizon Europe microbiome call, you should know about it. I am curious, you know, what can we do? You know, we could brainstorm right now. How much time do we have, Eloise, left?
-2 minutes.
Oh, well, okay. So we're out of time. So maybe let's, how can you join? Let's brainstorm, get together. Oh, looking at, I got some more pledges here. I like this. So yes, we have been brainstorming, and we think it's a really important avenue. The EU funding, we need US and Canadian funding, and other South Atlantic funding to basically make some of these UN decade things. There's a POGO, biomolecular observatories UN decade call for action pending. There's a Marine Life 2030 UN decade call for action, and all of those are concepts and frameworks. Without money, what are we going to do? I will give it back to, I guess, Margaret to close us out. I just want to thank everybody. And I'm still looking for, to the panelists. Is that let me see if that was the bios. Aha. I'm going to put this to everybody. Because where do we go from here? Margaret, I'm going to give it back to close this out.
Okay, I guess what we would be very happy is if everybody here present would subscribe to the Marine Forum. Send us there is an email there on that website, www.marinemicrobiome.org. Please send us questions. We are interested. We do want to promote marine microbiome. We do want to promote cooperations and collaborations. And we actually do want to extend this group into something bigger than it is at the moment. And it is for these types of things where there are research calls coming out and the opportunities, to network and to make introductions to folk and that type of thing. So we are interested in your thoughts, not not simply just on this call, but greater than this Horizon 2020, Horizon Europe call, we do want to figure out how to go about these things. How can we stimulate interest? How can we stimulate networking? How can we stimulate collaborations? How can we work together? And at the end of the day, how can we also make an impact on, let's say, the public awareness and consciousness about the marine microbiome? We all have our own national jurisdictions and things and stuff like that, and we have different ways in which we can work. And we just really would like to hear your thoughts on how we could go about these things. So please subscribe, please send some emails to us, and I think that's the very first step. Certainly there are already some Horizon 2020 projects that are still out there, like Atlantico, I think Triatlas is still around, iAtlantic, and a few other like Mission Atlantic on seabed mapping. Please also reach out to them. But specifically on the marine microbiome, come to our website, subscribe, and send us, there is a generic email there, but it is watched. People do monitor it, and we will bring it to our next meetings, and we'll see where to go from there. Okay, so I'd just like to thank everybody for coming here today. For a lot of Europeans, it's really late in the evening for them, and I really appreciate their staying power. For all of you from the west coast of the Americas, thank you also for participating, because it was incredibly early in the morning, and the rest of you for giving up your lunches to come here today and to participate in this wonderful event. We do want to see you all and work with you all in the future. You're just delighted to have you here. Last word, www.marinemicrobiome.org. Thank you, Eloise, for all of your help today. And thank you also to Sigurður Björnsson and Líður Erelsson. Thank you so much.
Yeah.
And the panelists.
All right. Thank you, everybody, taking the time to come. If you've heard anything you like, reach out to anybody that you see here. We're all incredibly friendly, and the worst you'll get is a conversation. So just thank you for coming.
Thank you. Cheerio. Bye-bye.
Bye.
Bye.
Recording.